So, Jacob, let's talk about biodistribution and some of the work that you're doing in that space. So, do you want to give a rundown of how whole brain imaging can be used to study biodistribution?
Yeah. So funnily enough, it was kind of the first thing that we actually did with whole brain imaging, because like 15, 20 years ago, it wasn't actually possible to label the whole brain with antibodies. So the options we had was to kind of label a compound or a drug or protein with a fluorophore and then inject it into a mouse and then just, let it distribute for a couple of hours and then take out the brain and then see where did it go. So the very first thing we ever did with that type of imaging was actually on a drug called Liraglutide, which was the predecessor of kind of Ozempic and Semaglutide. So it was like the first version 1 of the GLP-1 drugs. And at that time, it was used in the clinic for treating diabetes because there's a lot of receptors of GLP-1 receptors on the beta cells. So when you give GLP-1, it binds to the receptor and then causes insulin secretion, and that's why it's used. But then there was a paper that came out that you could actually use GLP-1 to label all the beta cells in the mouse. So we had one of the chemists at Novo making this fluorescent GLP-1 molecule, and then we took out the pancreas and we could see like all the beta cells lighting up. And then of course it became interesting to look in the brain as well, because at that time, people knew that it was reducing food intake and lowering body weight. So we started taking out the brain and to start to look at where did it go in the brain? Did it go to the right neurons that we knew was controlling appetite? And then obviously it became very interesting to compare different peptides where you knew they were some of them were more efficacious, like semaglutide. So did that distribute differently in the brain? So that was actually the first thing we ever did. And then you could say there were lots of other applications you could do using similar stuff. So people look at kind of a blood-brain barrier, like antibodies. Can you make antibodies cross the blood-brain barrier? You can look at antibodies that label kind of fenestrations. So you have some places in the brain where you have access from the periphery. You can label those regions with the certain antibodies, labeling those cells. But you can also just look at albumin and other biologicals. How do they distribute? How do they get into the brain? So it has a certain number of very interesting applications. And basically what you do is you just inject your compound and take out the brain and you look where did it go.
Yeah, so there's a lot that can be visualized with it, and we'll touch a bit more on the blood-brain barrier a bit later on in the conversation. But you've talked a bit about some of the targets that you can visualize there, but what about the quantification side? How does that work?
So because you can say in this case, it's like you label your drug with a molecule or fluorescent molecule, right? And you see where it goes. So typically the readout that we will use is fluorescent intensity. So we will basically segment out all the anatomical regions, but then we would measure kind of what is the fluorescent intensity in that brain region in a control group and a treated group. And then there is some kind of challenges because when you inject something like many, many millions, billions of molecules with a fluorophore, the tissue actually does become more fluorescent. So if you just have a PBS control, it will have lower intensity. So you have to kind of think that into the experiment. And I think also what we also learned is, especially for studying peptides, kind of the best controls you can do is basically have a knockout where you know that the peptide cannot bind the receptor. And then you see how does that then look. And then you can very precisely compare kind of the same dose, but then you can see in the mouse that didn't have the receptor, this region didn't label because the receptor is not there, but you still have the drug circulating and everything. That was really nice. But the main readout that we can do is fluorescent intensity.
Fantastic. And you mentioned about sort of drug targets here, but what can biodistribution be used for? What is it for?
Yeah, so I think if you think specifically about drugs, where we have the most experience is with peptides and antibodies. Peptides are super nice because they're usually quite small. You can make them in a machine and just you get your fluorescent peptide out. Of course, one of the things you have to be careful with peptides is whether they can still bind the receptor after you added the fluorophore to the molecule. So there will have to be some kind of assay development done to test that you still have the binding. And then the smaller your drug is, the bigger the fluorophore will be in comparison to the actual drug. So if you think about small molecule, for example, then they might be way smaller than the fluorescent molecule that you put on, which makes it a little bit, of course you can still have molecules that are working, but you probably change the properties of the molecule a lot when you put on a big fluorophore. And some of these fluorophores are known to be very hydrophobic and hydrophilic, so they can interfere with the distribution. So you have to work under the assumption that something is happening to the molecule. You just don't know what it is, so you have to set up the proper controls and study design to control for these things. But what we have done for peptides is we found that it was super useful for studying GLP-1. GLP-1 has a very easy biology. There's one receptor, one peptide, everything is easy. We have looked at other peptides where you have multiple receptors, or as soon as you start designing drugs that target, like polyagonist drugs that target multiple receptors, then it also becomes way more tricky to set up the controls right.
Yeah.
But then if you take antibodies, for example, they're much, much bigger. And here you can relatively easy add the fluorophores without interfering with the basically properties of the antibody. And I think also what we do find is that in some cases where you have therapeutic antibodies that you plan to use in humans, you would usually put them in a human backbone. So the antibody that you're injecting is actually a human antibody, and that you can stain for with other antibodies. So then basically you don't have to label it. You just inject it in and see how does it distribute. Then the last thing we have been working on is RNA and DNA molecules. And they are more tricky because there we don't know what controls, like let's say for a peptide, you know that it's supposed to bind a receptor in a certain place. A siRNA molecule can go into basically any cell. There's no kind of, we don't know the exact mechanism, but that also makes it more difficult to control where it goes and set up the proper kind of study design to look at whether you have some impact by the fluorophore. So kind of one example I can give is if you have a peptide, you can basically, a fluorescent peptide, you can pre-treat the mice with a non-labeled peptide, and you can in theory block away kind of the fluorescent signaling because the receptor is already occupied by a non-labeled peptide. That you cannot do for a siRNA or DNA molecule because the mechanisms that makes it go in are just general, so they will just bind whatever. So what we do there is we basically just look all over the brain, where does it distribute, and then we try to add different types of fluorophores, so like one that is very hydrophilic, one that's very hydrophobic, and then we see, okay, does the pattern look the same? And if it does, then we assume that it's at least not the fluorophore driving the distribution, then it's more likely to be the RNA or DNA molecule.
And supposedly the RNA, DNA molecules much, much smaller. And how does that impact like fluorescent and the ability to quantify the signal?
So in theory, you can say like one nucleotide is small, but the RNA DNA molecule can also be much bigger. So they can have like very different sizes and lengths. And also they have complicated secondary structures that you also don't really know. They can fold up on themselves, right? So there's a lot about the biology with these molecules that is, I think, slightly more tricky and less understood than an antibody or a peptide.
So is this a newer emerging technique, this one, like more recently?
Yeah, so I think there's a lot, obviously there's a lot of gene therapy that is aimed at knocking down genes in certain places. So definitely, but it has a lot of traction, and I think that it's really an emerging field. So, there are other ways you can think about then measuring kind of the efficacy of your drug. So if you're targeting a certain RNA molecule, you can actually measure whether that molecule is then knocked down in the brain by looking at the target rather than the actual compound. So yeah.
Yeah, that's fascinating. Can only imagine the implications of using that for like viral vectors and those kind of studying how those are working. And then the other thing here, talking about delivery, is the blood-brain barrier. You did mention it briefly earlier, but how does that fit into the picture here?
Yeah, so you can say in general, it's very, very hard for large molecules to cross the blood-brain barrier. They're not supposed to go into the brain. So there are certain places called the circumventricular organs, like CVOs, where you have access. That's in the hindbrain and the area postrema and the hypothalamus around the median eminence and arcuate nucleus. And then you have a few other brain regions where you have this on purpose leaky blood-brain barrier where your drug can kind of get in. And we know in those regions you have a lot of neurons that sense kind of what's happening in the periphery. But if you want to treat Alzheimer, for example, or Parkinson, where the pathology is spread out all over the brain, it's very, very hard to get your drug to those places. And so there's recently been a lot of development in developing brain shuttles, which is basically antibodies or other molecules that binds to transferrin or other transporters in the blood cells or in the vasculature. And that's basically a little pump. So when they bind that, they're pumped across the blood-brain barrier and get into the parenchyma on the other side. Yeah. And when you put these shuttles on your antibodies, the impact on the distribution is just amazing. So you can completely change kind of how the drug distribute in the brain by adding these little shuttles. And I think there are now clinical trials running with Alzheimer drugs that have these shuttles on, and they're looking super promising. So this is definitely one area that I personally find super, super interesting, and where there's just like, science has really come together to solve this really complicated question, and now it works. It's just amazing.
That is amazing. It's so exciting as well.
There's a lot of companies working in defining new shuttles that can be used. And one other maybe observation that we have made in terms of getting across the BBB is that so what we saw in the old days when we worked on GLP-1, then the way you extend the half-life of these molecules is adding certain fatty acids to the peptide that makes them bind, for example, albumin or HDL or LDL in the blood. And what we saw was that sometimes you would see the peptide go to places that couldn't be explained just by PK or normal kind of levels in the blood. So we have this, something we've never showed, but there is kind of this assumption that basically by binding to these plasma proteins, because those plasma proteins are then being transported into the brain, you can say they're kind of piggybacking on some of these plasma proteins to access kind of certain places in the brain.
It's really interesting. And then how can it be used to study target engagement?
Yeah, so you can say this is one of the really critical aspects of looking at drugs, like, because you want your drug to go to a certain place, but you also want it to function at that, whenever it gets to that place. And that could be to see whether it binds the receptor. So the thing I mentioned before, where you try to see, okay, so where do you have a signal? Where do you have your receptor? Where do you then have a signal of your drug? And if you remove the signal or the receptor, do you then lose the signal? So those things are, I think, in my view, quite solid evidence that you actually target a receptor in a certain place in the brain. Then we do have other readouts where we can look at, so normally when you bind a drug to a receptor, for example, you activate a cascade inside a cell. So typically that would lead to some kind of phosphorylation or different pathways being activated. So if we have antibodies that can recognize some of these downstream effects, that's also a very, very nice readout of where you actually activate the receptor. But this is slightly more tricky because it's it relies on available antibodies that you have for certain pathways. Then the last thing we have done is to look at, where, for example, in Alzheimer's disease, if you have the pathology, you can kind of measure whether your antibody is binding the pathology on the other side of the blood-brain barrier, right? And here we try to kind of measure whether, the simplest question is, do my antibody go to that place and do we see a signal? If yes, then if I have my shuttle, can you then get away with using lower doses of your drug and then get the same kind of engagement as you would without the shuttle? So those are the type of endpoints that we typically try to quantify to see whether the drug actually hits the target that it's supposed to.
And say that I was running a drug discovery project, how would this accelerate it?
Yeah, so obviously you can say knowing how your drug distributes in the brain is super important for many. So most of the side effects you typically have with drugs happens in the brain. So whatever nausea you might have from GLP-1 drugs or anxiety or, other kind of unpleasant side effects is usually because you activate unwanted population of neurons in the brain. So you can say there's twofold. You want to know where it goes in the brain. And of course, you also want to know, do I target cells that I'm not interested in targeting? And we've previously discussed CFOS that could also be used as a readout of off-target effects, but for biodistribution. If you just look at visualizing, you can say, if I go back to the example with GLP-1, we know that there's a lot of GLP-1 receptor in the brain. Maybe you with your drugs, only target 10%. And of course, that's super important to know if you then try to develop a new molecule that you want to target different types of neurons in the brain, that you can actually see early on. Like if you make 10 compounds that you want to test, you want to pick the one that hits the most neurons. Then you would run this study and then select the ones that gives you the most signal, or a signal in the places that you want.
Absolutely. Thank you. And has this been used in pharmacology before?
Yeah, so it's been used a lot, I think, in obesity. So I think I also mentioned, like, for looking at GLP-1s like Ozempic, Wegovy, but also other molecules like Tirzepatide. So a lot of these peptides have used this technology to kind of see where they go, what type of neurons do they engage. And then I think recently for the shuttles, it's turned out to be super, super nice to show kind of whether the brain shuttles affect biodistribution. And sometimes it's like, you don't need that many mice. When science really works, you can get away with very low study groups and numbers because it's just so clean and the effects are really dramatic. And that, I think, has been the case for looking at blood-brain barrier transport. Then you can say for siRNA and other molecules, AAV virus, it's also turned out to be quite important to understand, like, if you have an AAV, like, where does it go? Especially because there's some toxicity involved with developing AAV viruses. So there are certain aspects of this where it's super important to understand the distribution early on. And then, of course, a mouse is not a human, so it can never stand alone, but it can give you useful insight early on in your drug discovery program.
Absolutely. And in terms of my theoretical drug discovery program, what data do I get from you from this service?
So typically we would calculate the fluorescent intensity, right? And then we would typically have a control group, maybe a number of groups with different peptides, different fluorophores, and then we will provide statistical analysis in all brain regions, right? So we'll provide these kind of statistical maps where we look at average maps of a whole group. I think one of the important things with this technology is because you can overlay many maps from the same group, you can get this kind of statistical representation. We know that if you inject a molecule, some of it will be bound in vasculature, some of it will, be in certain places that is irrelevant for kind of understanding the mode of action. But when you do these statistical maps, you get very, very high statistical evidence for where your drug is exactly the same across all the animals in the group. So one example could be, let's say you have a peptide stuck in a blood vessel that will never be in the same place in 2 mice because it'll probably be stuck in different places. But wherever you target and engage the receptor of the drug will always be in the same place. So this statistical way of looking at it is really, really useful and very precise and gives you a very good understanding of exactly where your drug is working. And all of these intensities you then get in plots, and you can look them up and see in 3D and 2D how it distributes. And you can also, I think one of the nice features that we have in this virtual brain where you get your data, the CNS-Voyager, is that you can actually look up and see where are the receptors of the drug, because you can look up the virtual expression. I know that's mRNA level, so it might not reflect all the projections, but at least you could say where you have your receptor, you should also have your drug accumulating.
So you can save, assumingly you can save a lot of time and effort having that mapping done for you as well. They're like having that all done in one place, aligned against the atlas, and the information's there.
Yeah, and I think some of the programs I have been involved in, I think this is extremely useful information to have when you have to go out. You have to apply it to IND or NDA to kind of qualify your drug. This data has actually been used. The GLP-1 data we did for Novo was used to qualify Saxenda for an NDA for the first obesity drug, Saxenda. Yeah.
Wow, that's very impressive. And talking of the Voyager platform more broadly, like what does that have to offer? You talked about the visualization there, but what are some of the other features?
Yeah, so I think previously what we've seen is that the ability to overlay these statistical maps is also what can drive your decision making, right? So because let's say you compare 5 different compounds and you suspect they distribute differently, but you don't know where, you don't know exactly how they distribute differently. So then you just make the statistical maps for all of them. That actually allows you to very precisely say, okay, they're either all the same, which then kills your assumption. But if you see some of them distributing differently, you could take the 2 extreme and you can compare what is then the efficacy of these 2 drugs. So I think for decision making, I think it is very, very important to understand where your drug is going, what kind of potential effects could that have. It could also be like from a, if it's a side effect profile you're looking for, then that could help you close down that, project early on, which is sometimes in the pharma world equally important to starting up new things, that you kill stuff early if they're not showing what you want them to show.
Yeah, and a bit of a side question perhaps, but how would scientists normally go around using this data, or if they want to use your service, like what is the normal workflow for like working with this data? What's like the manual version look like?
Yeah, I think normally, people, obviously PET has been used a lot to study, like you put a radio label on your compound, you do like a PET scan, but the resolution there in PET imaging, especially in mouse or preclinical work, is really, really low. So basically, even when you have a signal, you don't know exactly where it is. I wouldn't say, quite often PET and light sheet imaging can go quite well in parallel because you're basically using exactly the same kind of approach. You have something, you inject it in, you look at where it goes. And we have done studies where we actually had evidence from both PET and light sheet, and when they support each other, that's super nice. So that's one thing you could say. Another alternative is, of course, cutting up the brain and look. I think what is important here is then quite often when you cut up the brain, you focus on certain brain regions. So you basically, if you're looking for side effects, you have to make some assumptions that, okay, I want to look in the hippocampus. You look at the hippocampus, but you miss everything else, right? And this is where the whole brain, like you get like the complete understanding of where all your signal, everywhere your drug goes, which is I think for these types of studies, super important.
So that's more like traditional histopathology then, or immunohistochemistry,
exactly.
when you're talking about the slicing there.
And I think, of course, now we're talking a lot about acute effects, right? Where you inject something and you see where it goes. But it could also be like you want to do more chronic studies where you look what happens to the... So what we know quite often with drugs that target certain receptors is that they will actually lead to a downregulation of the receptor itself over time. So it's like desensitizing the system. So there's something also interesting in looking at, okay, so you have an acute study, where's my drug going? But if I treat continuously for a week, does that then change kind of the distribution of the drug because there are no longer receptors expressed in certain places? So these are typically the type of studies that you would do.
Fantastic. Well, thank you very much, Jacob.
Cool.